Okay, so I think we can start with the next session. Audio and recording and video are okay. And so thank you for joining in the afternoon as well. I think this morning you had a very nice example of how we moved from that very simple pattern, the Berger's wave recorded on a piece of paper, to these complex models, I would say, that take into account the spatial dynamics of the EEG on the scalp, as well as the relationship with the structure and with our internal model of the outside world. Now we are going to move to another fundamental aspect of EEG research and research in general, which concerns more methodological considerations. And to introduce the next two speakers, I would say that one of the typical approaches that we have in the study of EEG, but in general, is to what I call to look for norms. Basically, we focus on a specific brain features, like let's say alpha power, and then we focus on a specific task or population, and we try to find whether there is a difference in that specific feature. So very simple relationships in a way. And in doing so, we often average the brain feature of many participants in one group or in one condition. And in this way, we neglect the high inter-individual variability that may be intrinsic to any brain feature. And also, we often encounter the fact that many different lab researchers can use different features, can focus on different aspects of brain and of the brain behavior relationships. And this ultimately leads to some very important methodological considerations, which is the focus of the two presentations you will attend next. The first one is by Dario Gordillo-Lopez, and then we will have Melissa Faggella. And here you have the information about the content of their topic. And I would just start by introducing the first speaker, Dario Gordillo. And with his biography. So Dario received his degree in acoustic engineering in 2015 in Ecuador. In 2017, he moved to Barcelona, Spain, where he obtained his master's degree in neuroscience. He started his PhD at EPFL in 2019 under the supervision of Professor Michael Herzog, where he investigated neurophysiological and contextual visual processing impairments in schizophrenia. Dario currently is a postdoctoral researcher at the Charité University Hospital in Berlin, and he works in the lab of Professor Peter Hulas, investigating visual processing impairments in patients with schizophrenia and working on the development of OHPM MAG for translational research. And I leave the floor to you.
Yeah. So good afternoon to everyone. I don't know if it's okay if I proceed without the mic for the people a bit further. I don't speak very loud. So yeah, first of all, thank you, David, Maria, and all the people that contribute with the organization of this symposium. Thank you a lot for the invitation. So yes, David mentioned I did my PhD here in Lausanne at EPFL, and to pay a tribute to my former lab, I will introduce some of the results that they obtained, and I will do so by also introducing what's the classic approach in schizophrenia research. So generally, those starts with a paradigm. For example, here you would need to declare whether this lower bar is tilted to the left or to the right. Right here, right would be the answer, and this is very easy. But then, if you introduce a mask, then the task becomes very challenging, right? So patients with schizophrenia were tested with this paradigm, as well as controls. And what it has been observed is that patients are very impaired with this paradigm. So essentially, they need 100 milliseconds more of time to be able to process the target. So if you think about it, if you need 100 milliseconds more to perform certain computation, that can signify a lot of impairments. So then they have looked at what happens in the brain. So this impairment, this behavioral impairment, was also associated with reduced amplitudes in the brain. So this is measured with EEG. And you can here see that patients have less brain amplitudes around 200 milliseconds. And this was associated with some top-down mechanism of enhancement of information. So again, with these results in hand, there is a theory proposing that the deficit is not necessarily about visual processing. So like lower level visual processing is relatively intact, but there is a problem when patients need to enhance this faint information. And this is proposed to happen through deficits in the cholinergic system or in recurrent processing in general. So further studies were carried out, and then they were able to find a correlate at the genetic level, which explains all these differences. So this is a really nice way to do science, and we have learned a lot about schizophrenia using this approach. However, if you think about schizophrenia, it's a very complex illness that is associated with a lot of impairments, not only in visual processing, but also in memory processing, for example. There are well-established deficits in working memory. Also in more structural aspects of the brain, like white matter. Also with some paradigms using EEG, like measuring evoked responses or resting state. Even immunological impairments, and even at the level of skin responses, patients show impairments. So naturally, there comes the question, like, how do these abnormalities relate with each other, right? Like, if you think about schizophrenia being the cause of all these impairments, then you would expect that there are statistical dependencies between these measures. And there are not many studies which have done that. Actually, there are very few studies that have asked this question, whether impairments, deficits are correlated with each other. So here we did not test very disparate domains, but we zoomed into neurophysiology, and we asked the question whether there are correlations between neurophysiological impairments in schizophrenia. So we use resting state EEG, and this is a really versatile tool to investigate schizophrenia, because with this data set, we could probe different mechanisms. At least it has been shown in with some methods. So it's really the same data. So you process the data differently, and you tap into different processes. So for example, classic way to analyze the data would be to do a spectral analysis. So you look at the power at different frequency bands. Then you would compare controls to patients. And here, for example, you can see that patients are not the same as controls. So they have enhanced power, for example, here in the theta band. So this is for illustration, but this is real data. And often it has been shown that, for example, theta band power is impaired in the patients. So patients exhibit higher theta band power. And this has been related with impairments, for example, in dopaminergic function, or abnormalities in other higher frequencies has been associated with hyperexcitability in the cortex. So these results are often used to favor certain theories about schizophrenia. So there are other methods, for example, looking at how sensors or how different sources are related, for example, using connectivity. And these methods have been used to analyze patients with schizophrenia, and it has been shown that patients have increased connectivity at different frequency bands. And this was related, for example, with neural inhibition, and also decreased connectivity at other frequencies has been shown to relate with attentional processing. And other well-established methods, such as EEG microstates, for example, have been also shown to be impaired in patients, and this was related with attentional control or genetic risk, among many other methods that I'm not showing. And it's not only that there are differences between controls and patients, but also these differences tend to be associated with large effect sizes. For example, here I am illustrating the example of EEG microstates, and you can find effect sizes of 0.80 something. So this is a high effect size. So if you look at the distribution of effect sizes in fields like neuroscience or psychology, this is not very common to have a D prime of 0.8. So these are large effect sizes, very large effect sizes. So we analyzed the data of 121 patients with schizophrenia and 75 healthy controls. And we applied the methods that I showed you before, and also many others. So we look at the literature. We found methods that were applied to analyze data of patients, and we employed these methods, and also some others which were not which were used in for study other populations. So through all these methods that we applied, we obtained 194 EEG features. So one feature would be the power in the alpha band, for example. So the next step is with each feature, we compared patients to controls, and then we use the correlations to understand the relationship between these methods. So we found that 69 out of 194 EEG features showed significant group differences between patients and controls. And the coincidences range from 0.5 to 1. So these are mid to large effect sizes, which correspond to discriminabilities of 60 to 70% between controls and patients. Just to illustrate, we found very different features, such as, for example, if we are in an oscillation symposium. So this is a specific feature about alpha oscillations, such as the self-similarity across different time windows. So this is often estimated with the trended fluctuation exponents. And we found that patients have less the trended fluctuation exponents, like lower values compared to controls. And this is a result that was published in 2012, like the first paper, and we replicate these findings. So patients have decreased the trended fluctuation exponents in the alpha band. Also, other aspects of the oscillation, such as the distribution of bursts in the beta band, for example, also showed a difference between patients and controls. And again, I am putting there the effect size, like the Cohen Z, and these are large effect sizes generally. This is like other methods, such as connectivity in the theta band, also network properties from EEG connectivity matrices, EEG microstates. We also use methods such as the entropy, so like the complexity of the signal. Also, we use these cross-frequency coupling methods to analyze like interactions between lower and higher frequencies. Also methods of time variability, among some others. So this is just to illustrate some examples of methods that delivered significant differences between controls and patients. So we have these methods, and then we know that these correspond to alterations at the neurophysiological level in patients. So here, when the next step is that we correlated these methods, and essentially what we ask is whether, for example, a patient that has a higher value, for example, in the theta band connectivity, would have also a high duration of the microstate C, which both have been related with schizophrenia and both point to really critical theories about the illness. So from the outset, we expected that they should correlate, right? However, what we found is that there are relatively low correlations. So there are actually low correlations between all these features, right? So this that I'm presenting here is a correlation matrix. So what you have here are the names of the features that deliver the significant difference between controls and patients. And the colors here illustrate the magnitude of the correlation. So pale colors here would indicate low correlations, whereas more strong colors indicate high correlations. So you would see that the matrix is rather pale. And if we look at how is the distribution of these correlations, we see that the median correlation is 0.1, which is very low. And this was the case for Pearson correlations. Also, we tried Spearman correlations and also other methods of like multivariate or nonlinear correlations, for example, based on vector distances, like distance correlations. And results are quite similar. So most of the correlations are rather weak. Well, there are also a few correlations which are high, but generally, you would expect that if these methods deliver a significant result and they are able to classify controls for patients, namely they point to critical aspects of schizophrenia, you'd expect that they explain more than just what the feature tells you, right? But this challenge a bit this idea of how much explanatory power we have with one feature. And yeah, this that I am showing here is determines what is a strong correlation. So we see we are far from from that. So what this could tell us is, for example, if you analyze your data here with using a method like microstates, for example, you would find that these are two example patients. You would see that in this case, for example, they would have lower values compared to controls. And then you analyze this with another method, right? And then you have patient 2 in this case that is very much impaired. And then you change your analysis and now you find a different result. And every time you find a different significant result, then you will have very different results compared to other methods. So this can be problematic if you think that the idea of using these methods is to be able to reduce this complexity and to point to the neurophysiological core mechanisms, right? And this lack of correlation is it kind of goes against this goal, because every time you use a method, you will point to a different mechanism. So this can be problematic if we want to point to the core aspects of schizophrenia. Now there is the question, obviously, why do we find these significant or group differences and low correlations? And would say, yeah, it could be that the tests are not, or the methods are not very good, right? Like the reliability. However, we found that while most of the correlations are low, we find some strong correlations, right? This could indicate that if a method can correlate with other methods, it should correlate, right? So it's not entirely uncorrelated. So indicating that there is a signal that we are picking up. And also there have been previous studies showing that some EEG features have adequate reliabilities, like the EEG microstates or the trended fluctuation exponents, the spectral amplitudes and entropies, among some others. And also we have, I will present some preliminary data that we have some of these same patients and controls that took part in this first experiment, in this first recording, also came to the lab again after 5 years. So we re-recorded the resting EEG, and we analyzed how stable is this feature, how stable were these EEG features. And here it's 82 instead of 69 because we had a bit larger sample size. So, but this doesn't matter for now. So what I want to say is that it's true that some of the features had rather poor long-term stability, but there were many features which had like above 0.4 intracast correlation, which is considered a fair reliability. And also some had good reliability, and this is quite surprising because this is 5 years apart. And there were features that had really excellent reliability, so above 0.75. So it's not entirely explained by measurement noise. Another idea why we would have significant group differences and low correlations could be that what we consider large effects are not necessarily large. For example, we would have, that I'm illustrating here, two distributions with a Cohen's d of 0.8. So as I said before, it's very rare that, it's rather rare to find a D prime that it's above 0.8. So you would consider that this is a large effect. But if you look closely, right? So here you select an optimal decision criterion, and then you would see that a lot of the cases are not explained. So you have a D prime of 0.8, but still there are some patients which are not impaired at all. And you have many controls which have very high values. And for example, for a Cohen's d of 0.8, the discriminability is 65%, in the sense that 35% of the patients don't show the effect. And you would say, yeah, it could be measurement noise, but I'm showing that it's not measurement noise because these measures are very stable, right? So there is something more. There is true variability behind. Now, if we want to achieve 80% of discriminability, we would need Cohen's d of 1.68, which it's very rare in the fields. Maybe in physics you find something like that, but in psychology, I think it's very rare. And just to mention all the effect sizes in other fields, so we also find that they are not necessarily much larger than what we find in EEG. For example, measures like the thickness of the brain or like structures in the white matter. Still, you know, these methods can tell a lot about the brain, but they are not able to classify very well patients from controls. Another possibility why we have significant group differences and low correlations. Could be like the idiosyncrasy of the measures, right? So we know that with EEG, we expect to target certain mechanisms related with some processes. In this case would be schizophrenia, but there are many other factors that also contribute to the variability of the data. So for example, would be aspects related with comorbidities or something that comes after the process, after the mechanism that we measure, some byproducts, let's say, of the target mechanism. So in that sense, when we have a significant result, we explain that we expect that all this variability relates with what we want to measure, in this case, something crucial or like fundamental about schizophrenia, but there is also another aspects that contribute to the variance, and this can compromise the correlations. And finally, it could also be that this reflects the heterogeneity of schizophrenia, right? So in this sense, like low correlations would indicate that there are multiple underlying factors that contribute to the disorder. And then we could ask the question whether all this indicates that there are different causes for the same disorder. There are some results indicating that it's true that schizophrenia can have different causes, right? And then if we think about heterogeneity, we can ask questions like what type of disorder is schizophrenia? So I'm here illustrating the case of schizophrenia, but you can think about any complex research situation, right? Like you said, what type of disorder is schizophrenia? Is it like an additive disorder in the sense that if you have more impairments in more features, it would lead to worse outcome? Or is it combinatorial in the sense that you need just specific combinations of features that would increase significantly the risk? Just as an example, we know that there are certain variants in the genes that really increase significantly the risk for schizophrenia. And then you just need one variant and you have a really dramatic increase in the risk. Whereas there is also this idea that there is this polygenic nature of schizophrenia, and you need a lot of variants which contribute a little bit, and they lead to the same outcome. So this is interesting because different cases could be explained by different models. And in any case, if there is true complexity, and in this complex scenario, then I would argue that using one feature will be limited, right? If we want to understand generally, like really the most fundamental aspects of schizophrenia. And just to say, it's not that we discover something totally new. So people have thought long time about it. And there was recently a really nice paper from Faggella where they put forward some interesting arguments about how we should approach complexity if we want to understand complex disorders like schizophrenia. For example, they propose that there are many assumptions that we implicitly make, but we don't want to make, but we implicitly make them, right? If you do a t-test, for example, you are implicitly assuming that the variability is noise, right? Or that there is one model that explains all the data. And if you ask someone, you will say, no, I don't think that. But with the methods you use, you imply that to some extent, right? And they propose there are ways in which you could, other methods, like for example, they propose using normative modeling, for example, to account for heterogeneity. There are other methods, but in any case, what we propose is that within subject studies can be really informative about complexity. And there are some other assumptions, such as there are brain regions that operate as isolated units instead of networks. So we still try to find the one part of the brain that explains it all, but we know that this is not the right thing to do, or that there is a one-to-one mapping between what happens in the brain and what happens at the symptomatic level, right? So we need to think a bit differently if we want to go in this direction. So more and more people are kind of taking seriously, let's say, this variability. So to conclude, analyzing multiple features can help identify contributing factors to complex disorders. There is not a single factor that explains neurophysiological deficits in schizophrenia, right? So we need more than one feature if we really want to explain neurophysiology. And I like to say this because it happens all the time that when I present people say, yeah, of course, no one thinks that you can explain schizophrenia with one method. Yeah, who would believe that? But you say, yeah, I agree. But then if you see how the research is conducted and how the results are reported, they are implicitly assuming that you can do that. So within subject studies can help understanding complexity. So again, I was showing that normative modeling could be one way, but this requires a lot of data. But there might be other strategies that you could use, like for example, carefully obtaining different measures for the same subjects, see how stable they are, or see how they relate with each other. So being more like smaller samples, but with better data quality, for example. And again, yeah, low correlations might indicate multiple causes. And this is also in line with some interesting results that they have shown that there are these clusters of patients which share similar traits. For example, there are patients which have intact like white matter or gray matter structures, but they have other deficits which are more functional. Whereas you have this other group of patients which have very, very strong deficits in the structure of the brain. So there might be different subgroups, right? And then each of them requires a different type of methods or different type of features would explain better their symptoms, for example. But in any case, I want to mention that both approaches, like really to zoom in into the mechanisms and also to look at how they relate with other mechanisms, both approaches are needed and are crucial if we want to understand complex disorders like schizophrenia. So yeah, finally, I would like to thank my collaborators, Janir, like the Eka Maya, who collected the data, Andreas Brand, and Michael Herzog for his support. And thank you very much.
Thank you. Let's see if we have questions.
Very, very interesting. So I have a bunch of questions. I'll rattle them off and you can do what you want. First off, what do you observe in the healthy controls in terms of these features? What is the clinical characterization of the patients? Are they all chronic? Are they early phase? How do you deal with medication effects? How do you know when they're at rest that they're resting in the same way? And why not look at domains where the variability is potentially lower? And the spontaneous EEG is renowned for being highly variable. So you're kind of starting off from we expect a lot of mess. Whereas if you look at, we didn't mention auditory stimulus impairments. There's a lot less variability. There are animal models. There's pharmacological characterization. So can you comment on some of those points?
Yeah. So first, what we observe in healthy controls, this is something that we found also striking, and we want to learn more about it because if you look at the pattern of correlations, it's very similar. And this is striking because it seems that there is not a lot of signal that is present in the patients that is not in the controls, right? So this struck us a lot actually, and especially when we find the stability. And again, we have some good D primes, but still, again, there are many controls which have the impairment, but this is very stable. So we want to learn more what happens with, for example, we are now doing an analysis where we account how many deficits, for example, controls have. And for example, how this relates, for example, with cognitive symptoms and so on, because I think there is this idea that some healthy subjects that show high genetic risk also show certain impairments. So we definitely need to look at the controls. And I think not many people look at it because they focus more on the clinical groups, but I think we can learn a lot from a healthy control that has impairments that are like schizophrenia-like. Then about the resting state, it's true, but actually the main studies that motivate us to do this were some studies that came out in around 2006, where they analyzed these electrophysiological methods, actually, like the first correlation analysis that was done in schizophrenia. I think it was where they correlated the P50 mismatch negativity and I think P3. And they also find lower correlations in these domains. Also, there are some studies showing, although cognitive factors explain are a bit more related, but there are other domains in which you also find low correlations. And I would argue that in resting state, you expect more correlations because it's the same data, right? Although I agree that again, going now to your next question about the instructions, I think this is also critical, like, because I think here the participants were asked to relax during 5 minutes and not fall asleep. But this could be different if you ask to mind wander, for example. But still, there are many studies which have used different instructions, but in many cases, the features are stable, right? And I would assume if you have a long enough recording, they could do a lot of things and they will be averaged out. It's just my intuition, but maybe it's not true. But I think it's crucial. I know many people are now using the questionnaires, like what were you doing during rest? And then they try to see if this explains also variance. So yeah. And sorry, I think I'm missing one other. Try to address them all. Yeah. No, thank you for the question.
Thanks.
Do we have other questions online? Okay, then I think. Thank you again, Dario. And we can move to the next speaker, which is Melissa Faggella. She studied life science engineering and neuroscience at EPFL in Lausanne. And she is currently a neuroscience PhD student in EPFL Laboratory for Psychophysics under the supervision of Professor Michael Davis. And she's working on schizophrenia patients, longitudinal EEG data, and individual difference. The floor is yours.
Thank you. So good afternoon, everyone. It's a great pleasure for me to be here to share with you some of my work on individual differences in task-related EEG data. So let's dive right in with some general comments. So in biomedicine, and notably in neuroimaging, we can safely say that today the vast majority of research is not done on single individuals, but instead we tend to aggregate many subjects together and to draw conclusions on the group average. And this is particularly true for task-related EEG data, as most studies focus on deriving the group average waveform, so the event-related potential, and then on comparing the amplitude and latency of few landmarks of interests across conditions or across groups. Like, for example, here we would focus on the N1. And we use the average because we want to derive general principles that applies to the whole population, but also because individual data often presents a lot of inter-individual variability. And this variability acts as a nuisance when we are trying to derive clear-cut scientific principles. But as harmless as the group average seems, it comes with some assumptions. First, we are assuming that the average is a meaningful summary measure that improves the signal-to-noise ratio. Then we also assume that the conclusions made at the group level naturally generalize back to single individuals. And then we also assume that the individual differences that we are averaging out are irrelevant to the final conclusions. But today I would like to show you, to challenge a bit those assumptions, and I will do so by showing you some individual level longitudinal data of a visual task called visual backward masking. My colleague already introduced it, but wait. Cannot move. Ah, good. So in this task, we present participants with a stimulus called a vernier. It's 2 bars presented like this. And the lower bar is either shifted to the left or to the right. And the task of the participant is to report the direction of this offset using a pair of handheld buttons. And it's not as easy as it seems because the target is only shown very briefly for a few tens of milliseconds. And is quickly followed by a bulky stimulus called a mask. And the mask is here to disrupt the integration of the target. And in fact, the shorter the time between the target and the mask, the harder the task becomes. And when we want to measure the behavioral performance to this task, we let this time vary. We call this stimulus onset asynchrony, SOA. And a good performer would have a short SOE, for example. So this is a demonstration to show you what the task looks like. Let's have a small audience attention test. Please focus at the center of the screen. Okay, so if you perceive that the lower bar was to the left, you have good attention, so keep up the good work. So this task was also adapted for EEG experimentation. So we fixed this time the durations of the three screens to be able to repeat many trials and do EEG. The task of the participants remains the same. They still report the offset direction. And we have two control conditions. We have one where only the target is present, one where only the mask is present, but we don't tell the participants that there is no target. And we have two regular visual backward masking conditions. One is harder because there is a short SOA, and one is a bit easier. And we record in total 180 trials per condition, and the 4 conditions were mixed within blocks. Okay, so just for the context, but my colleague also introduced it before, we have been interested in this task for a long time now, and we gained interest for it because there is a large behavioral and electrophysiological group difference between schizophrenia patients and healthy controls in red and black. And interestingly, there is also a milder deficit in the first-degree relatives of the patients in blue here. And as schizophrenia is heritable, the milder deficit of the relatives suggests that this task is related to the genetics of the disease. So this is why one of the reasons that makes this task interesting. And so because of all those properties, almost 15 years ago, a longitudinal study was launched. And we tested schizophrenia patients, relatives, and healthy controls. And luckily, a subset of those participants agreed to come back to be retested 5 years later for the EEG paradigm. And then we even have a subset of people that came a third time, again 5 years later. And this is a quite unusual long time interval for a longitudinal study. Here you have the current sample sizes. And so the original goal of this study was just to demonstrate the stability of the marker. But what I want to show you today is some unexpected results that we found while looking at individual level data. So just 2 words about the 2 main methods I will show you today. One of the methods we used for the EEG is called global field power. It's a summary measure that quantifies the total amount over all recording electrodes. And if you look from the mathematical point of view, it is the spatial standard deviation across electrodes. And you can see an example here on the right of how the individual electrodes in the rainbow colors give rise to the GFP waveform in green. And one of the main advantages of GFP is that it is a kind of dimensionality reduction method that allows you to consider the impact of all recording electrodes instantaneously instead of focusing on one single electrode like we will do in a classical event-related potential analysis. And the second method I used is the microstate analysis. So for those of you who are not familiar with this technique, the term microstate refers to a semi-stable topography or pattern that stays around for a few milliseconds to up to 1 second in EEG data. And some researchers hypothesize that microstates represent basic building blocks of brain processing. And to obtain the sequence of microstates of a given snippet of EEG data, either resting state or ERP, it's the same procedure. You start by applying clustering on the instantaneous topographies, which means that for your piece of data, you want to find the most common maps. And so you end up with a few template maps that we call the microstates. And then what you do at the end is that you backfit the template maps back to your data by doing competitive backfitting. So for each time point in your data, you look in the template maps for the most similar one. And then the way results are usually shown is the GFP waveform. So for example here, and the colored areas correspond to the different template maps. And then in the end, what people do is that they simply compare either the statistics or the sequence of the maps across groups or across conditions. And so global field power allowed us to condense EEG across the spatial dimensions, across the electrodes, while the microstate analysis allows us to reduce the EEG along the time dimensions. So we can see that those two methods kind of complement one another and give us a global view of what is happening with the EEG data. So let's move on to some results. First, some group level global field power results that we obtained by averaging the individual GFP waveforms of subjects. We do that by group. And here I show the first testing group average for the 4 conditions. So each plot is one condition. And each color is a different group. It's the same color code as before. And those cross-sectional results perfectly replicate what we showed in previous studies. Namely, we find this nice proeminent GFP peak at 200 milliseconds after the onset of the target. We call this N1. And importantly, we also find this group difference in N1 amplitudes across patients, relatives, and controls. And yeah, so let's now look whether those group level results hold after 5 years. And here I just add the second testing with a dashed line style, and you can see that they perfectly overlap. So everything is very stable after 5 years. And for all 3 groups, for all conditions. So it's a good news for the marker, the trait marker. It seems very robust. And so subsequently, also the lab proposed a neural mechanism to explain this electrophysiological group difference. And the story I told so far is quite similar as we will do in a task-related EEG publication. We average across subjects, we find a group difference, and then we build a story around this group difference. But it is implicitly assumed that the group level waveform generalize back to the individuals, and also that the averaging helps us getting rid of individual level noise. But so, okay, let's check whether this is really noise, random noise. So in this slide, I show each plot is a different subject, so the GFP waveform of individual subjects, and they are all performing the same condition. Here I only show the first testing, so the blue waveforms. And as a reminder, I show the group average here on the side for the controls. And as you can see, the inter-individual variability is quite remarkable. Subjects differ a lot from one another and also strongly differ from the population average, with differences in number, amplitude, and latencies of peaks. And now the most interesting part, let's see what happens to those individual differences when we retest the same people after 5 years and 10 years. And so we can see that it's not random noise because it's conserved after 10 years. And in other words, we can say that the inter-individual variability is not caused by irrelevant factors, such as fatigue, stress level, caffeine, medication intake, because there are also some patients among those waveforms. It's also not problems due to the data acquisition, because all those factors I just mentioned, they vary across testing, so they cannot explain this. And also remember that we are considering global field power and not individual electrodes. So it's not just one electrode that is stable, but rather the global activity pattern over all recording electrodes that is stable after 10 years. And the fact that the individual differences remain so stable after such a long time suggests that they do carry meaningful information about the subjects. And I will discuss some hypotheses a bit later, but first let me show you some more results. Yeah, as I said, I want to complement the GFP results with another method, so the microstates. And in the literature, what is typically done is to average all subjects from a group, and then you derive the template maps for this group. And so this is what I show, the template maps at the top for the group. And in the middle, you can see the backfit for 3 subjects. And here I only show one testing. But what I did instead is to do the analysis for each subject individually. So we have one set of template maps per subject. And most interestingly, if we do this for each testing, so each column is one person, and we have the 3 retests, the 3 rows are the 3 retests So we can see that people conserve their maps, their template maps, and their time course also. And in addition to that, there is high inter-individual between subjects in terms of maps and time course. And this reinforces the results we found for the GFP. And additionally, it also confirms that it's not just one electrode or one landmark peak that matters, but rather the whole time course of the ERP and the whole topographies and the sequence of the topographies that remain stable within individuals. And before we move on to the discussion, I would like to share two more GFP results. First, here I show, again, one subject per plot, but this time the different colors correspond to the 4 different experimental conditions. And what I want to highlight here is that the group average differences between conditions are not necessarily reflected in all individuals. So at the group level, we found that the mask conditions had kind of a truncated peak. The N1 was not very present in the mask condition in green here. While when we look at individuals, not all of them present this pattern, and some even have almost no difference between conditions, like subject 1 and subject 2 have almost no differences between the conditions. So it seems that maybe the group level effect was driven by a few subjects. Yeah. And I think that the fact that the between condition pattern of variability present at the group level is not reflected in around half of the participants, I think it raises some concerns about the generalizability of the group level conclusions. And the last result I want to share concerns the pairs of siblings. So we have pairs of parents, brothers and sisters, and children and parents. So that means they have 50% of DNA in common. And in each of those plots, I show one pair. So the red waveform corresponds to a schizophrenia patient, and the blue waveform corresponds to one brother or sister. And as you can see, there is a great resemblance between the waveforms of siblings, and overall they have comparable number of peaks, amplitudes, and latencies. And these results suggest that the individual differences are to some extent inherited and are thus caused by something we are born with rather than by something we learn through experience, maybe something cognitive. Okay, so let's now summarize a bit everything I showed. We saw that event-related potentials are stable within individuals over very significant period of times, so up to 10 years, and that there is also a lot of inter-individual variability. And finally, we saw that the EEG of related individuals are quite similar, which suggests that EEG is heritable. And importantly, those results are not just true for one single electrode or one single time point, like a landmark, but rather extend to the whole duration of the ERP. So here we had 400 milliseconds. And it also remains true across all electrodes as supported by the GFP and the microstate results. And while searching through the literature, I found that similar observations had been already made as early as the 60s. And also it had been concluded for a wide variety of tasks, ranging from very low-level tasks such as light flashes, but also more sophisticated tasks like go-no-go tasks, for example. And in this first figure on the left, Dustman and all, for example, they exposed 4 subjects, so each plot is one subject, to light flashes weekly for a few months. And you can see that the individual level waveforms remain completely stable over weeks. In this second figure in the middle, the same authors, they showed light flashes to pairs of monozygotic twins, so 100% of DNA in common. And each plot again is one pair of twins, and compared to our results, we can see that the ERPs are even more similar. So this is a really interesting study. And lastly, so far I only talked about ERPs, but it seems that very similar results have been made for resting state data. It was more about the statistics, but the inter-individual variability and intra-individual stability seems to hold true also for some parameters. For example, I found some studies superimposing the power spectrum of twins, and we can also see that they are perfectly identical. And another study that I found that illustrate well the inter-individual variability and irritability of resting state EEG is the last figure on the right here. And in this experiment, one professor printed on individual sheets of paper few seconds of 8 electrode resting state EEG data for 114 pairs of monozygotic twins. So here, for example, he shows the sheet of paper of twin 1 and twin 2. And he scrambled the sheets of paper. And then the goal of the experiment was just to match by visual inspection the pairs. And on the first try, he managed to match 95 out of the 114 pairs. So it seems that there is really a lot of similarity in the resting state data of twins. And even though it has been known for a long time that individual differences are stable, so far I haven't found many works investigating really the information content of those individual differences. So I came up with my own hypothesis. So in my opinion, the fact that individual differences are stable over such long time intervals and are inherited rather than acquired through experience suggests that it might be caused by an anatomical rather than a cognitive factor. And possible factors previously mentioned in the literature include individual differences in scalp, skull thickness, surface area, and localizations of brain regions, for example, V1, or the white matter distribution, for example. But my personal main suspect is the pattern of cortical folding, because it doesn't change much over time. It is highly heritable within pairs of monozygotic twins also. And thirdly, there is a high inter-individual variability. For example, in this study, an expert manually labeled the folds of 20 brains. And here you can see the superimposition of the 20 labeled brains. And you can see that the same sulcus can be in very different places. So there is high inter-individual variability. And I also think that the folding pattern is an interesting candidate because of the properties of the electrical fields captured by the EEG. So depending on the distance or orientation of the source, the EEG captured at the level of the scalp changes dramatically. And here, I think it is well illustrated in this simulation study. So a bit more precisely, the further away from a source you are, the weaker the signal will be. So for example, if the source is at the bottom of a sulcus, it will be much weaker than if you are at the top of a gyrus. And additionally, if the signal, so the signal that is perpendicular to the scalp are much stronger than the signal that are parallel to the scalp, like for example, in the wall of a sulcus. So, okay, yeah, this is my hypothesis. If you have other ideas, I'm really happy to hear about them during the questions. Before I conclude, maybe these are the take-home messages from today. We saw that individual differences in event-related potentials are stable across time, up to 10 years, across pairs of siblings. And that then those differences probably contain relevant information about the subject, maybe about its anatomy. And finally, all these results, I think they kind of question whether group averaging is always the best solution, and maybe we should do stratification. Maybe this is an idea, or other I think other methods are being developed at the moment. And yeah, with that, I want to thank my PI, Professor Michael Herzog, my colleague Dario, and our two collaborators from Georgia that collected the data. So thank you for your attention.
Thank you, Melissa. And do we have questions?
You gave me instructions. Thank you very much. Absolutely fascinating. The question I would have is about the topographic clustering and backfill.
Mm-hmm.
So what you showed, if I understood rightly, is you're applying that clustering either to the group average and then to individual subjects, or it's a different individual subject.
Yeah, so the way they do usually the analysis is to compute really the group average ERP and then do the clustering on the group average ERP.
Right, but your result.
Me,
Backfitting, by the way, is testing whether it's consistent across individuals. right? You're getting a pattern in the group average. You're then testing, is that pattern similar across subjects?
Now here, what I do is really, I do the analysis once for each subject and each testing, one individual analysis for each testing for each individual. And this is just to show that the same maps and the same time course is captured after 5 and 10 years. It's just another way to show the stability.
That shows stability across time.
Yeah.
But if I understood you right, you were making a claim about variability across subjects.
Yeah, because they have different time course and different template maps.
Yeah, but the template maps by definition, that's the starting conditions of the cluster. You're not making any constraint on whether those maps should or shouldn't be the same across individuals. What would be more interesting is to apply the clustering on subject 1 and test if it's observed in subject 2.
But for example, I would have expected at the peak, at the main landmark of interest at 200 milliseconds to have maybe the same map almost in most people. If
it's, let's say, an N1 topography, it should be an N1 topography across individuals. But that is what the standard clustering and backfitting is testing. Is in the group average data, there's an N1 map, let's say, and is it the case that my participants indeed exhibit an N1 map? That's what methods are testing. By mathematically, that I can assure you, there's.
I'm not sure to follow, but I'm
You're
curious to
calculating
hear
the
more.
spatial dissimilarity between a template map from the group average and a template map or the actual data from an individual subject. And you're asking what is the spatial correlation between those? And is it best for one map versus other maps? So by definition, the backfitting is testing consistency across individuals. What you can do is you don't need to use a group average. You could use, is David the standard human being brain response, and do we all look like David? I could also do it for you and David and ask, who do I resemble more? Do I resemble David more, or do I resemble you more?
So a bit of fingerprinting in a way.
Yeah, the methods allow for it. The GFP, I agree, is different, but it's measuring a different different aspect of in this case, the event-related potential. It's just how much the variation in space is. And that, of course, varies across. That doesn't surprise me in the slide. But so topographic clustering shouldn't vary. I mean, if it's robust statistically, it means it's not variable, or it's less variable across them.
So you are suggesting instead of backfitting maps that are estimated on the group, so start from the assumption that everyone contributed to this map.
You can. I mean, there's nothing in the method that says you have to start from a group average.
It's just the way it's usually done.
And we've done it with Michael. We've done single trial and single subject topographic, and it's quite interesting that it is consistent across the group. Yeah, but I think the topography is actually far more consistent than the strength.
Yeah.
I guess was my main point.
Yeah, I agree.
Do we have other questions?
Just out of curiosity, but the difference pattern, let's say, of the GFP of the zero potential is linked with the performance in doing the test of the subject. So, for instance, one subject had a really good performance, so all the trials, you use all the trials to have the average GFP, so it's why maybe we see a really clear peaks or not.
So far, I really tried to find if there is maybe a kind of typical answer of the good performance, but no, it seems that the pattern of GFP and the performance is completely unrelated.
And in characterizing this GFP and also in the pattern of your recordings, I think your structure hypothesis connect to the validness, but also maybe you also check, I don't know, the impedances, of the recording set. It depends on the pattern, of course, of that itself. If there's some.
Yeah, this is also something I want to look into. Yeah.
Okay. Thank you.
Okay, I think we can, no, please, another one.
Yeah, thank you so much. I think my question is related. So how much does this EEG variability is relevant for behavior? Does it also explains behavioral variability or not? Is it different? If not, how much is it relevant in general? And maybe when are you have you investigated?
So far, I haven't found any link between the pattern of electrophysiology and the performance, behavioral performance. But I think this has a lot of importance just behind just the anatomical and individual differences, just because when you do the group average, you are erasing out all this inter-individual variability. And maybe this is meaningful. And maybe also by averaging out super heterogeneous waveforms, you are creating spurious signal that doesn't really represent anything. So this is one of the reasons it's really important to understand what's behind these differences.
Maybe I ask the last one. So your hypothesis of the cortical folding, which makes perfect sense. But then what do we learn from this?
So there are cases where I think it would matter. Like, for example, I saw some publications that suggest schizophrenia patients have some cortical folding abnormalities. So maybe some of the group level group differences that we find could be related to this. So in some cases, it could be useful. And I also think that maybe if we find that it's really the case and that cortical folding has a big impact, maybe we can develop methods to remove this kind of artifact. If we want to consider it as uninteresting and remove it as an artifact, then maybe we could really improve the signal to noise ratio of EEG data.
Yeah.
We show that across different conditions, the waveforms change. But then if there is a cognitive aspect, it cannot be only the folding, because then why would different conditions are different?
But then why have we subjects where there is no difference between the conditions, like subject 1 and 2? The question goes direction, I think.
Okay.
Maybe, yeah, maybe.
So there might be a component problem.
It could be, for example, that the region of interest is hidden deep into a sulcus for some subjects and is quite accessible in some other. Maybe, for example.
Maybe just to support you there. So Cheryl Ain, back in the early 90s, mid 90s, did some work with Steve Hilliard on exactly this role of alkaline folding, in particular on the visual potential.
Yeah.
And there you do find, I mean, if you're doing retinopathy, we're very retinopathic mapping with EEG. So it does vary, but then on top of it, when you ask those participants to do, I don't know, an attention task at a given location, of course you see modulation of the event-horizon potential that varies across individuals.
Yeah.
Which also then varies depending on their cortical folding. So the two coexist perfectly. I don't think it's at all a problem.